Okay, so one of the things we've always wanted to do with the FIRE team is make sure that we could expand it to include other researchers that we think could add something to what we're working on and the work that's going on. And again, this is one of those things where we thank everyone in the audience who's been helping us raise funds and keeping our coffers filled so we can do things like this. Because of you, we've been able to add a fifth member to our FIRE team. And so today you're going to get to hear from Dr. Kevin Nash. He is also at the University of Florida, so he and Ed can work together. And you heard a little bit today from South Florida? University of South Florida, sorry. Sorry to football fans in the area. And you heard today also about some of the work that they're already doing with Agilis. So I'm not going to take any more time, and we're going to turn it over to Kevin Nash.
Okay. Can everyone hear me? I'd also like to thank the FAST team for actually inviting me to join the FIRE team. And I'm really looking forward to working on the project that I have, and I'm really looking forward to working with the rest of the FIRE team as well. Since I haven't been here before, and most of you wouldn't know me, I thought I would do an introduction of where I come from. I graduated at the University of Queensland, so I am Australian, go Australia people. And then from there, after I graduated with my Ph.D., I moved on to the University of Florida, where I worked for a while. And there I worked on adeno-associated virus. And what I was particularly interested in there was actually the replication of this virus. So I was looking at the biology of the virus. But while I was looking at the biology of the virus, I actually became interested in the virus as a tool for gene therapy. And in particular, I became interested in the tool for neurological diseases. And I actually started collaborating with other researchers at the University of South Florida, looking at Alzheimer's disease and Parkinson's disease. And then from there, I actually moved down to the University of South Florida to continue my collaborations. And since then, since I've been at the University of South Florida, I've continued my work on Alzheimer's and Parkinson's disease, but I also started collaborating with Dr. Weber, looking at Angelman Syndrome. So I just wanted to point out some of the advantages of using this viral vector adeno-associated virus. Now, it's particularly important virus because it is non-pathogenic. So this virus, the wild-type virus that occurs in nature, does not cause any disease in humans. So you're not going to cause any problems by injecting this particular virus, even if it was a wild-type virus. You don't get a huge immune response, which is particularly important because some of the viral vectors that have been used have caused an immune response. And some of the immune responses have actually caused death in some of the studies. You can infect non-dividing cells and dividing cells with this virus, which is particularly important because we're interested in infecting neurons. We have what we call episomal maintenance, and Greg talked about this earlier. The virus does not integrate into the genome. So it remains episomal, so it remains outside of the genome, so you're less likely to integrate your DNA into cells and then maybe cause cancer to develop. You get long-term expression. So a lot of studies have looked at how long you can actually produce the protein from the gene that you're injecting. And there's a lot of studies now that have shown very long-term expression. If we look at mice or rats, you'll have expression for the entire life of the mouse. Some other studies in dogs have seen up to 6 years without any change in the level of expression. We don't have any of the viral genes present. So this is important because if you have viral genes present, you may get an immune response to those particular genes. And because they're no longer present, we're unlikely we're going to cause an immune response to the virus that we're injecting. And of course, there's the virus itself, and the production methods for the virus is well characterized. And OK, here we go. So if we take another look at the virus here, it's a very simple virus. It's what's called a dependent-virus, in that it actually doesn't replicate by itself. It needs other viruses to infect a cell in order for it to replicate. So it's a very basic virus. You can see here, it's a very simple genome. It only has essentially 2 proteins present. And we can take those particular proteins and we can remove them. So like I said, we don't have any of those proteins present in the vector that we're injecting into the animals. And then we replace them with the gene of interest. In this particular case, we're replacing it with the UB3A gene. We can also change what we call the promoter. And that's what's driving the expression of the gene. So we can actually replace that with different promoters depending on where we want to target the gene of interest. And in this case, I'm just talking about a tag. Sometimes we tag the proteins in order to be able to see them easily. So when we inject our virus, we can specifically target the protein that we're making in the cell because it has the small tag on it. So if you can imagine, here's a picture of the virus. If you can imagine, the AAV is like a soccer ball. And if you normally would see the viral DNA inside that soccer ball, okay? But we essentially remove that DNA and we're replacing it with the DNA that we're interested in. So now we have a way to inject that DNA into the cells we're interested in. I like the long pauses you get when you push the button. Okay, so here's an example of an injection of the AAV virus. This is actually AAV9. So Greg was talking earlier how there's different serotypes of this virus. Essentially what that means is the protein that makes up the outside of the virus is slightly different. So there's small mutations within the protein sequence. So they actually become immunologically different. So they can, in fact, infect different cells differently. But here I just have an example of how well we can infect the hippocampus of a mouse. So this was a single injection on one side. And you can see clearly, looking at sections throughout the entire brain, we can target pretty much the whole hippocampus. And this was important for the first study we did. And a lot of people have talked about this. Jen Daly's work. We used this method to inject the UB3A gene, and this case was the mouse UB3A gene, into the AS mice. And we got nice expression of the UB3A gene in the hippocampus. And then we also saw restoration of some of the behavioral deficits and also rescue of LTP. So here you can see a wild type animal. When you stimulate, it maintains the stimulation. In the green down here is the AS mouse, where it doesn't have the UB3A gene, so it can't maintain the stimulation. Oh, sorry about that. And then here where we treated with the virus, you can see that we're maintaining that level of LTP stimulation. But one of the problems with the virus, of course, is it is limited to the site of injection. And if you remember when I injected in the hippocampus, you could clearly see that I was only in the hippocampus. I wasn't throughout the entire brain. So AS is not limited to one part of the brain. So we need to find ways to achieve more global distribution. So we want to get this protein therapy to the entire brain. So some of the things that I'm interested in is looking for alternative methods to distribute the virus or distribute the protein throughout the entire brain. So one of those methods that I'm looking at is using the ventricular system. So the ventricular system makes the spinal fluid essentially within your brain. And so you have this spinal fluid system within your brain, and it pretty much, essentially you can imagine that it coats your brain. So you have these ventricles here, and the system actually floats around and it goes down into the spinal cord as well. So one of the serotypes that we have is an AAV4. And it's interesting because AAV4 can actually infect the cells that line this system. So what we can do is inject the virus into these ventricles, and then we can use these ventricles to express the protein we're interested in, and then try to deliver that protein to the rest of the brain. So we can use these cells as like a small factory to produce protein, to try and deliver that protein to the cells that need it. And I'm not sure how clear you can see this, but here we have an AS mouse, and hopefully you can see that there's not a lot of staining along the edge here, which is the cells that line these ventricles. And then here you can see this dark staining here, where I've injected an AAV4 virus that makes this UB3A protein. And so what we're going to be looking at is whether we can use these cells to produce the protein and then restore function within the mice. Another serotype that we're looking at, this is actually from another group. They used serotype AAV9, and then they essentially injected it into the ventricles as well, but using a targeting down here, and it's called intrathecal injection. So you still end up in this spinal fluid, but this virus has the ability to actually pass out of the spinal fluid and then infect cells within the brain. So you can see here they show that there's expression throughout the brain, and they're getting neurons and other cells targeted throughout the brain. So we may be able to use this system to have these particular hotspots, we could call them, or factories, to produce the protein again and deliver it to all the other cells within the brain. So we don't necessarily have to target all of the brains with all of the cells in the brain with the virus. We can target some of them and then have those produce enough protein for the rest of the cells. And then one of the last thing that I'm looking at is looking at pathways within the brain. So we know that the brain is not just different regions that don't talk to each other. They don't interconnect. Of course they interconnect. And so there's particular pathways that make different connections within the brain. And so I want to target some of these interconnecting pathways to see if I can make protein. I can infect a cell in this region here. It'll make protein and deliver it to another part of the brain. And I hope you can see here where I targeted this region of the brain here. What it's showing here is actually a green fluorescent protein and not the UB3A protein. So we typically use this as a control to set up and see how well it works. But you can see when I inject in this region here, I'm getting expression in many other regions of the brain. And so I'm getting spread from one particular region where the neurons branch out into other regions of the brain. So hopefully I can use this technique to infect these cells with UB3A. Then those cells will project, they'll deliver the protein to the other regions of the brain, and then hopefully those cells will take up the protein and then restore their normal function. And I didn't have much time to make my acknowledgement slide, but since I was a little busy this morning working on a grant, but I'd like to thank Ed Weber, Jen Daly, and a couple of members of my lab who are help working on this project at the moment, Christian and Mindy. Thank you for your time.